Hello! Today in this section I'll be walking through how to properly normalize a protein of interest using REVERT Total Protein Stain from LICORbio. This type of normalization will normalize a target of interest against the total protein signal in that lane. This method is often a preferred normalization choice of publication journals over housekeeping proteins, unless you plan to perform a Western blot against a post-translational modification. For the rationale behind using total protein stains for a normalization strategy, it would be best to review the earlier lessons in this course. Here I have the result of a Western blot experiment loaded into Image Studio Lite software. In this experiment, 10 micrograms of lysate was loaded into each lane. After transfer, the membrane was stained with REVERT Total Protein Stain, as shown as red here in the 700 channel from Odyssey Imager. Subsequently, the membrane was destained from REVERT, blocked, and then a Western blot was performed against protein BCL2. That is shown here in this image in green from the 800 channel of an Odyssey Imager. Now, so far I've already used the tools in the analysis ribbon. We've added rectangle shapes around each lane for the REVERT Total Protein Stain and each band for our BCL2 protein. To recap the features of how to quantify your target of interest, please make sure to review the lessons covering basic quantification. Now, once you've quantified your targets of interest in Image Studio Lite, we're going to want to go ahead and export our data to a graphing program of choice. Here, I've gone ahead and exported our data into Microsoft Excel. In Excel, the quantification from the REVERT Total Protein Stain is placed into one tab, and the quantification for our BCL2 is into another. In our normalization analysis, the first step we'll want to perform is to calculate a normalization factor. This normalization factor will be used to correct the calculated signal values for a BCL2 protein. To do this, we'll first want to examine the calculated signal values from our REVERT Total Protein Stain shown here. When you look at this value from lane to lane, we can see there is variability in the amount of protein that was transferred onto the membrane. This highlights the importance of performing normalization and the power of using a membrane stain like REVERT Total Protein Stain. As we examine the values, we're going to want to find the lane with the highest signal value. In this example, as we look from lane to lane, we can see that here in lane 1, this has the highest signal value across the entire membrane. Once you've gone ahead and located this value, you'll next want to go ahead and divide the other lanes by it. This can easily be done in Excel by typing in a formula where you go ahead and divide the adjacent lane by that value. So in this case, 218,000. Once you go ahead and type that in, we've now created a normalization factor for lane 1. That same formula can be applied to all lanes by easily copying and pasting it in Microsoft Excel. This calculated value is our normalization factor, which we can type into the table for easy organization. Now that we have this value, we can copy and paste it into the BCL2 tab to go ahead and then normalize that target. Now when you go ahead and copy this in Excel, make sure when you paste it into your next table that you paste the values and not the formulas. Now we'll use this normalization factor to go ahead and correct the signal value and generate a normalized signal. To do this, we'll go ahead and create a column for this. We'll then go ahead and create a formula where we divide our signal value by our normalization factor. When we go ahead and apply the formula, we'll now have a normalized signal for that lane, which we can go ahead and copy and paste across all lanes in Microsoft Excel. Now at this point, we have successfully used our REVERT Total Protein Stain to calculate a normalization factor. We then went ahead and used that normalization factor to correct our signal intensities of our target of interest to generate a normalized signal. Working forward, as we do further analysis of this target, we'll want to make sure we use this normalized signal and not the non-normalized signal shown here. Those are the basics of how to accurately and precisely normalize a target of interest using REVERT Total Protein Stain in Image Studio Lite and Microsoft Excel. If you want to learn more about Image Studio Lite or REVERT Total Protein Stain, please visit www.licor.com. Thanks for taking the time to watch these lessons, and good luck with your own analysis.